Please welcome Kerstin Haase, Experimental and Clinical Research Center, Charité Universitätsmedizin, Berlin. The cancer circulome.
Hello, everyone. I think by now you're all well aware how cancer arises, so through mutation and genomic alterations. But so far we've only discussed linear chromosomal DNA as a cause for cancer. And I now would like to use this time to introduce you to the circular DNA, extrachromosomal circular DNA in cancers. They've actually been described in childhood tumors as far back as the 60s, but surprisingly in the more than 50 years since then, our understanding as to their function and as to their frequency in cancer hasn't improved very much. So why am I interested in circular DNA? My area of research is tumor evolution, and one of the strong drivers for tumor evolution is intratumor heterogeneity, so meaning genetic differences among the cells of the same tumor. And circular DNA can contribute a lot to this because it doesn't have a defined way of segregating cell division. So while the linear DNA very nicely splits among the daughter cells, the circular DNA can, in the most extreme case, just completely accumulate in one of the two daughter cells during cell division. As a computational biologist, it's also a neat algorithmic challenge because we now have vast amounts of sequencing data that's just been mentioned in multiple talks, and all the algorithms we throw at them are also geared towards linear DNA. So now we have this data, and we also have data in there that we can already use to define our circular DNA, as in discordant read pairs, split reads, and now also the ultra-long read that we acquire through the new next generation accounts. So now we can integrate all of these and try to develop tools to define these circular RNAs from a sequencing sample. And last but not least, it's very important from an expression level, because these circular RNAs can change the expression level in a cell, not only just by aggregating these copies of oncogenes that are part of the circle. The circles have the capacity to integrate back into the linear genome, and then they either disrupt tumor suppressor genes or enhance other oncogenes and drive their expression. And when our lab looked at circular DNA in neuroblastoma patients, we've actually seen that patients that had circle-derived rearrangement had a worse prognosis than patients without these circle-derived rearrangements. So we hope that by further investigating circular DNA, we can hope to better identify what drives genomic rearrangement and remodeling in tumor biology. Thank you very much.